This movie depicts the rupture of malaria-infected erythrocytes at the completion of schizogony, the release of Merozoites into the extracellular fluid, and the invasion of other erythrocytes by these Merozoites. In a typical experiment, as shown by this sequence, normal and malaria-infected erythrocytes were inoculated into a Dvorak-Stotler controlled environment culture chamber. The chamber being inoculated here consists of a perfusible closed system in which it is possible to maintain steady-state physiologic conditions for protracted periods of time. The chamber is placed on the stage of an inverted microscope equipped with differential interference optics and observed with a 100x oil immersion objective. The microscope image is relayed into a low light level video camera. The resulting video signal is processed with image-enhancing electronics and recorded on videotape. Selected sequences were transferred to 16mm motion picture film by electron beam recording. All of the sequences depicted in this film are presented in real time. Here is an example of an erythrocyte containing a mature parasite, the schizont, just prior to rupture. Note the pigment containing residual body that appears as a dark refractal mass in this cell. The Merozoites are attached by their posterior end to the residual body. Vesicles have appeared on the erythrocyte cell surface. These vesicles disappear as the cell elongates and swells. Note the movement of individual Merozoites within this cell just prior to the sudden explosive rupture. Some Merozoites are free of the residual body at the time of rupture. This sequence shows two Merozoites attached to a dark refractal residual body forming the shape of a V. Note the structures at the end opposite to the residual body, which are probably the paired organelles. The movement of Merozoites is caused by several external forces, such as Brownian motion. In addition, Merozoites have intrinsic motility. These two Merozoites have settled on the glass surface and propel themselves along this surface. Note the marked deformation and displacement of the erythrocyte during invasion. Note the Merozoite attached to the dark refractal residual body. The orientation of this Merozoite during penetration of an erythrocyte can be clearly observed. The end that first enters the erythrocyte contains the paired organelles and is opposite to the residual body. After invasion, the residual body is apparently still attached to the Merozoite, but remains outside the erythrocyte. There is sudden swelling of the erythrocyte after penetration.